Microdose monthly webcast. We are at the end of March, heading towards December with the Garden States event, which I'll talk about in just a moment. Tonight I'm broadcasting from the lands of the Wurundjeri people of the Kulin Nation and would like to pay my respects to elders past and present, as well as anyone who may be tuned in today or is watching the video in the future. I'd also like to recognise that we benefit from the knowledge and insights of First Nations people here in Australia and from First Nations around the world, including their long relationships with the plants and fungi, the original technologies. If you know what country you are on, then I invite you now to drop that into the comments section, and if you're up for it, provide a brief introduction of yourself. Our comment sections are always a lively place full of knowledge and a good opportunity to connect with like-minded people. My name is Nick Wallace, and I am the co-founder of the Australian Psychedelic Society, currently head up the Melbourne chapter. I've also spent the past decade volunteering and working in harm reduction in various capacities, and I'm personally passionate about not just seeing an end to this drug war, an end to prohibition, but also seeing the growth of a connected and vibrant community, and helping to create those connections and foster that community. EGA provides opportunities for critical thinking and knowledge sharing at the meeting grounds between plants, fungi, mind and culture. EGA embraces the psychedelic in the broadest sense, and the science, culture, policy, and nurturing the grassroots community that holds all of this together. We are heading towards, as I mentioned, the end of the year, which will be the Garden States event. And I just want to take you to the EGA website quickly now. So here's the EGA website. Oh, there it is. And if you want to register your interest, just scroll down, click here to register, and that will open up a form where you can register your interest right there. Just type in the details. We are holding off on ticket sales until we see what happens during winter, just in case COVID has any more surprises for us. Also want to just mention that Nimbin Mardi Gras, which usually happens within the month, has been postponed. You might have heard that there have been some pretty serious floods in the northern New South Wales region and across Queensland. Some of the biggest floods, again, unfortunately, because it wasn't too long ago that there were big floods in that region. And we want to send our love and hope that everybody that's there, anybody that's tuned in today, is able to rebuild and that it hasn't been too destructive. And we know for some it has been incredibly destructive. But Mardi Gras will be going ahead 16th to 18th of September. And you can find out information. I'll get our mods to drop in their website there. But in the meantime, Mardi Gras also has a fantastic YouTube channel with lots of previous talks on there. So you can check out some of those as well. Also uploaded to the EGA YouTube channel, which you can see right here, a new video, Kane Barlow, an introduction to Psilocybin cubensis, and you can view that now. That was literally just added, so just head to the EGA YouTube channel, make sure that you are subscribed, and you'll be able to find that there. And finally, I just want to mention two podcasts. The Australian Psychedelic Society now have a podcast. You can find their website, psychedelicsociety.org.au, and the first one is with musician Ben Lee, who you might have heard some of his more recent music, but I personally knew him from way back in the day when I was in high school in the late 90s, early 2000s, and Ben Lee had Cigarettes Will Kill You, a couple of other hits, and they were the ones that stuck in my mind. But I got to catch up with Ben, who is a psychedelic enthusiast and somebody that I feel like is after my heart a little bit, because I got to say, a lot of his answers were on point, and I really agreed with a lot of what he had to say. You might not, would love to hear any discussion. So psychedelicsociety.org.au, hit podcast up the top there and you'll be able to find that. One more podcast as well, Symposia. Our friends over in the US have been producing, along with the New York magazine, a fantastic podcast series called Power Trip. So the season Power Trip uncovers the secrets and exposes the darkest corners of the psychedelic renaissance through a twisted, deeply personal tale at the intersection of mind, body, and control. So that is co-produced by New York Magazine and Symposia. It sounds fantastic. Kind of, if you're familiar with Radiolab, I'd put it sort of there. So it's really, really well produced. Very impressive. So please do check out those. Support the people that support the psychedelic community. And a couple more information things if you want to stay up to date with everything that EGA has to offer. entheogenesis.org/connect. That is where you can sign up for the newsletter. Signing up for the newsletter keeps you up to date with everything that EGA has to offer, including information about tickets for Garden States 2022, upcoming webcasts, and also some of the other projects that EGA has going on. The resources that EGA has to offer are fantastic. Do go and check those out at the EGA website. And I want to just run you through, I've been talking about this the last few webcasts, and it's still in development. It is going to be a beautiful project once it's finished. It will be a big information sheet on sort of similar to the TripSit chart, if you're familiar with the TripSit chart. And again, I'll ask if the moderators can drop in the TripSit link into the comments section. Go and check that out. That chart is all about understanding what could happen if you mix certain substances together. Certain substances go all right together. Certain substances are quite dangerous together, and a lot of people don't necessarily understand that. And that includes pharmaceuticals as well, although that's a whole broader territory that doesn't necessarily get fully explored in the TripSit chart, but it's very important. So EGA's chart will be similar to that, but with plants, and I'm going to flash a quick example of what it's looking like so far. This is what it's looking like so far. Of course, the final artwork will be beautiful once that is ready. Next month, for episode number 14, For the Love of Weed and Justice, celebrating 30 years of Mardi Gras, we will be speaking with the organisers of the Mardi Gras Protestival, hearing what they have on offer for us, and also, I suppose, what's been going on in the Northern Rivers region. And finally, Garden States, of course, Friday the 2nd until Sunday the 4th of December. A 3-day forum for cultivating psychedelic plants, knowledge, research, and community. And we would love to see you there. It will be an opportunity for everybody to catch up in person, which is much needed, I think. Remember, again, we are live tonight. Please drop your questions into the comment section. Our moderators will be there ready to answer them or pass them on for our Q&A later on in the program. Make sure that you are subscribed to the EGA channel and pass it on to your friends. Anybody that might be interested, do pass it on. We're aiming for 8,000 subscribers. We're at about 7,840 or something like that at the moment, and hoping to get to 10,000 by the end of the year, which will put EGA among the top psychedelic YouTube channels in the world. Make sure to also follow us on Facebook, Instagram, and Twitter. And if you're watching this back later, the conversation continues in the social world, so you can continue that conversation on the social media threads or engage with us in those sorts of ways. Also, just again, I'm actually wearing one tonight, an EGA, an ISWAS shirt. I believe they've still got some EGA t-shirts up for grabs at iswas.com.au. That's IZWOZ.com.au. And the profits from that will be donated to EGA. So for tonight's episode, why do many psilocybe species contain psilocybin? What is the role of psilocybin in nature? What's it there for? What are the ecological functions of these psychedelic substances? By surveying the genomes of the organisms that produce these substances, we can identify evolutionary trends and events that raise hypotheses about benefits provided by these substances, and by extension, this can suggest ecological niches in which to search for new compounds with similar roles. So we're going to be joined by Dr. Jason Slott a bit later in the program to hear about that. But first up, I have a pre-record from Alastair Mctaggart. Alastair is a research fellow from the Centre of Horticultural Science at the University of Queensland. He studies the evolution of fungal pathogens with a focus on rust and smut fungi. He has completed postdocs in Africa, America and Europe, and Alastair has started a new research direction for UQ, studying the biodiversity of native psychoactive mushrooms in Australia. This research will determine whether Australian magic mushrooms, Psilocybin species, have evolved unique genetic pathways for the production of psilocybin and confirm the endemicity, the localness of our native taxa. Alastair will establish a living collection to safeguard genetic diversity and provide a platform to research the applications of psilocybin for human health. And now, Alastair's video. Stick around, we'll have Dr. Jason Slott later, including a Q&A, and we thank you for being along with us tonight.
Hey, Alastair here, and I'd just like to say that I recognise that Australian Aboriginal people had knowledge of mushrooms as foods, medicines, and biomaterials. And since last we spoke, I've had dreams about what I should have told you and what I didn't tell you. And so we're going to cover a little bit of that. We're going to cover some new knowledge that I've had since we last spoke about new species in Australia, or I shouldn't say new species in Australia, maybe new discoveries in Australia, and then about how magic mushrooms will reproduce. And I couldn't help but put up a couple of photos of Psilocybe cubensis that have been fruiting around southeast Queensland over the last couple of months. And so what did keep me awake was thinking about everything I was telling you in the project, and I'll cover what the aims of the project are soon. But I didn't really talk about the life cycle of a mushroom and help you understand about why I'm working on different parts of the life cycle. So there's that classic delightful purple spore print of that every taxon in Psilocybe produces a purpley brownish spore print. You'll see right here is that black spore print produced by a Panaeolus. And pretty much from my knowledge, when I was studying mycology in America, anything that has a purple or black spore print technically doesn't have an amatoxin and is safe to eat. However, you do not want to be the biological exception. So best to always eat what you know. That's the spores under the microscope. I think you've all seen that before as well. But you haven't seen this before. And this is just what I want to briefly cover because I love life cycles and talking about sexual reproduction. So the thing that make mushrooms and everything in Basidiomycota so unique is that they, in every single cell, they have two nuclei, one from each parent. So each nucleus contains one copy of every chromosome, and Psilocybe cubensis has 13 chromosomes. So this is called dikaryotic, and it's just a condition of Basidiomycota. They spend most of their life cycle with two nuclei in each cell. These little clamp connections, which you may have heard of before, is one of the characteristics of mushrooms or Agaricomycetina. They maintain nuclear fidelity. So as that mycelium is growing, they produce a little tunnel for the second nucleus to move along, just so that those two nuclei can stay together. Then when you have that big hymenium or big mass of hyphae or mycelium and the right environmental conditions, whether it's rain and a piece of cow manure or a temperature drop, then you have production of a mushroom, the sexual organ. So it's all about sexual reproduction. And in each of these gills, you'll have a basidium, which is the location where the two nuclei fuse, which is just called karyogamy, if you've ever heard that term, to make a diploid nucleus. Once you've got that diploid, you can have meiosis and the production of recombinant nuclei that go into basidiospores. Then what I'm really interested in is how these basidiospores cross and how they meet up and decide to become a dikaryon again. So I'm going to answer that later. So it's kind of mate compatibility and mate recognition. And this is what I wish I'd told you last time. So I'm working with single basidiospores, the haploid stage, so something that only has one copy of every chromosome. And in Basidiomycota, when you've you working with haploids, you kind of have something that's a little bit different. With plant pathogens, when they're growing as a haploid, they might cause a really weak infection. Or the group of fungi called smuts, they're non-pathogenic as a haploid, and they don't become pathogenic until they become a diploid. So it's just really easy for genetics, but you'll see it if you've ever cultured mushrooms before from single basidiospores, you'll just see that the growth is really flimsy, weak looking, not robust. And so that's what I work with. And it's really likely that from a culture that I make, that it doesn't contain any psilocybin at all, and that you'd need to really get the knowledge down of what to cross that culture with to be able to produce a mushroom. And that's what the industry will eventually have to work out as well. So I just wanted to give a quick recap of the project. So it's just really, I can only focus on 2 mushrooms just because there's not enough hours in the day. So subs and cubes. I love this photo from Miss Keats. This is from Tassie, and I just love this little purple spore print here. So the question surrounding Psilocybe subaeruginosa is whether it is endemic to Australia and it has been distributed internationally. And to support that hypothesis, we already know that it was originally described from Australia from native national parks in New South Wales, South Australia, and Victoria, out of eucalyptus wood chips and out of grassy woodlands. So it's pretty convincing that it's probably native to Australia. Its sister taxon, Psilocybe cyanescens, was described 20 years later from Kew Botanic Gardens, which is that big multicultural melting pot of soils and plants from all across the world. And it grows in invasive locations in Europe, similar to cubes here actually, and also in invasive locations down the West Coast. And what kept me awake at night was that Psilocybe cyanescens, so this taxon in the Northern Hemisphere, had become one of the go-to recreational species. And also I'd started to see it appear in patents back in the noughties or whatever, for extraction of psilocybin and also treatment with psilocybin. So that taxon, Psilocybe subaeruginosa, and cubes, just because Psilocybe cubensis is so readily collected where I live and all up and down the east coast of Queensland. So really nailing whether this thing is an introduced taxon in Australia, which it most likely is, looking at how many times it's been introduced, and then maybe one day linking up with some other researchers who have more data and we can really start to explore the global population. We can look at whether the introduction is just associated with the movement of cows, or whether the introduction is an intentional thing that is linked to the distribution of spores globally. So many cool little questions there. So these are the key points, which I think I've gone through pretty clearly already. But yeah. Okay. And then the research questions, which I've covered last time. So really, you could break this all down. This is a population study of Psilocybe subaeruginosa and Psilocybe cubensis in Australia. And it has the advantage of establishing a massive haploid collection of magic mushrooms that will exist well after all of our deaths. And Ronnie just asked me a question before about how long these cultures can be stored for. And the answer is indefinitely. So they're stored in glycerol, which is cytotoxic at ambient temperature, but at minus 80, it's like a little cushion around that mycelium, and you can take it out hundreds of years later and just start growing a mushroom as if it had not been at minus 80. So that collection just becomes a resource to use in the future, hopefully. Well, that's the dream. Okay, and then you've also seen this slide before. I just want to say I rarely culture from diploids, so I don't really need to ever be sent mushrooms. In fact, even me collecting them is kind of an inconvenience now, just because there's so much effort to get into the herbarium, or to meet the conditions of my permit. I need to have heaps of people. I need to just have one person witness me destroy it by an autoclave, but I'm lazy. But so yeah, when I notice a mushroom is really a heavy blue stainer, I might take a diploid or a dikaryotic culture to keep that genotype without having to ever cross it again. So I'll do a surface sterilization of the stipe, and then we expect mycelium to grow out of that in a couple of days. Most of the time, though, because I'm working with the haploids, this is the process. So a streak, then isolating a single basidiospore and having that haploid growth. And really for everything, growing stuff in liquid, when you've got the equipment to keep things really sterile, my word, you can grow up vast quantities very quickly. So an update on the project. Thank you to people who have been sending me spores. I've got a slide to thank you personally later. At the end, or kind of nearing the end of the cubensis season up here, we're sitting on probably 7 populations, 56 haploid cultures. Subs, because the project started after sub-season, so really we'd only had the things that I'd collected, and I can only collect them up the range of Queensland. So the cube collection has come along quite nicely. Subs, we don't really have enough to start answering any questions yet, except for questions based on their genomes. So I've been sequencing genomes. I sequenced 10 genomes, which you saw in my last talk. I don't know if you remember, but the haploid genomes are so much easier to work with than the dikaryotic genomes. So I also realized I didn't explain this figure well enough. This is really an illustration of the diversity of Psilocybe cyanescens. So in these kind of satellite-looking things, these are called haplotype networks, and we've used ITS, which I possibly explained was the molecular barcode last time. So this kind of teal deal right here contains Weroroa, Subaruginosa, Macarorae, cyanescens, azurescens, and alani. And when something fits inside that circle, it means it's not differentiated by a parsimony informative character. So pretty much very, very, very similar ITS sequences. And this is a clue that a species rank taxonomy is not going to be based on molecular barcodes, but let's look at the population and start to really get as many informative characters as we can from across the genome to start looking at how populations are structured and whether there's, say, something from Queensland still outcrossing or sexually reproducing with something from Tasmania, or whether they warrant to be called different species. And then just up here, this is the cube section. So cubensis, natalensis, and subcubensis. This one's natalensis right here. This one actually always gets me thinking. Kane and I, ages ago, were talking about the center of origin of Psilocybe cubensis. And this little natalensis circle out here, which is a South African taxon or an African taxon that grows out of hippopotamus and elephant dung, I think. It makes me wonder about the population that must extend up through Africa and into the subcontinent on big mammal dung. I'd love to start to look beyond what the community is growing with Psilocybe cubensis and start really getting our hands on some native, naturally occurring Psilocybe cubensis. So those big circles are just saying, hey, something's going on here. If you think there's further diversity, so if you want to apply 1, 2, 3, 4, 5, 6 names to here, well, you need to unravel that diversity a little bit more. Hopefully I've explained that better than last time. So since we last met, I gave a talk for one of the mycology classes up here, and a student asked me whether Psilocybe cube, why does it only occur? Why do cubes only occur in cow farms? And I kind of answered really quickly with more confidence than I should have had that cubes must occur everywhere because they're dropping 2,000 spores every minute. And it's just that the niche of being in cow manure in a carbon or nitrogen-rich substrate is what's required as the kind of environmental cue to form that fruiting body. So what I did, I wanted to test it a little bit more because I thought, well, I can probably show this for realsies. And there's a big sequence database of soil from all across Australia collected by these guys, base essentially, where you can get any bacteria or fungal sequence from the soil to work out what's there. And I expected that we would see Psilocybe cubensis all through the sampling sites from New South Wales and Queensland. And oh my word, how wrong I was. So in this tree, the bold are the base sequences. So that just means that they're the ITS sequences that were sequenced by base from national parks in Australia. And when we look up in our cubensis clade, There's not a single cubensis sequence from over 3,000 sites in Australia, which is really, really interesting. It might not be the be-all and end-all, because all across Victoria, New South Wales, and South Australia, they haven't picked up a subaeruginosa sequence, but still, it's kind of telling. Like we do get subs in very undisturbed native areas. It's just so amazing that there's no cubes in any of those soil samples. So what I would suggest that these bold sequences are probably natives to Australia, just because of the sampling strategy of Base, who really went to natural undisturbed areas. And this is the one that excited me the most, this Litchfield National Park and Kakadu National Park. So these sequences are represented by more than 100 sequences, we're talking out of a couple of thousand. So it's definitely, there is a mushroom that is a species of psilocybe. And we know that nearly all species of psilocybe produce psilocybin. They all have purple spores or purpley brown spores. So that's just part of being in that monophyletic group. You have those characters, the 4 to 5 genes that produce psilocybin. So there's a mushroom in the Northern Territory, possibly in Queensland, and it's related to Psilocybe youngensis. And one of the taxa described by Gaston Guzman is Psilocybe bruniocystidiata, which he described from Fagaceae wood chips in Papua New Guinea back in 1972. And he speculated that this taxon was sister to Psilocybe youngensis. So even without a physical specimen, just based on sequences, I just found it fascinating that this one from the Northern Territory is sister to youngensis. And I imagine that if you're living in the Northern Territory or far north Queensland and you look on some wood, decaying wood, you're probably looking for something like this, which is just that classic psilocybe habit. So yeah, it'd be great if you got a spore print, anyone gets a spore print, and we can look about determining whether this is a new taxon or not. The last little update that I have for you is just about sexual reproduction. So these are 2 haploid individuals, and this line where they meet, where you can see it starts to get robust. So it goes from kind of wispy, whimsical hyphae into something that's fluffy and white. Whereas over here, you kind of had this interaction zone where not much is going on. It's not quite meant to be. So what is causing that? And how can we learn from this? How can we predict who's going to cross with who if we wanted to start breeding species of psilocybe? So mating in the Basidiomycota, I kind of feel like you probably don't want me to get into this, but it's regulated just at two loci. So there's things called homeodomain genes. You could kind of think of that like an X and a Y chromosome. And then they've got pheromone receptor genes. So genes that are just saying, hey, we recognize each other. So they're controlling, yeah, we know each other, and yeah, we're compatible. And a lot of the time, all of them must differ to cross. Now, if everything has to differ to cross, and they're in completely different parts of the genome, the homeodomain locus and the pheromone receptor locus, or think of them as genes, if they're separated and they all must differ, it's almost like having an additional 2 genders. So you've got, and this is called tetrapolar mating. So you have to differ at 2 places to have a compatible sexual relationship. Whereas with bipolar mating, you only have to differ across one kind of locus. So that's if the pheromone and homeodomain genes are really close to each other, or the pheromone genes are no longer involved with mate compatibility and they're just associated with mate recognition. Now, that may seem like a lot to take in, and I'm very mindful of that. You could just think that in Basidiomycota, some are like humans, where there's just the equivalent of an X and a Y, and some, we don't know which, have an extra 2 genders. And the reason that they have that is to stop inbreeding, so that because spores don't have legs and they're on the whim of the breeze, they might end up near their offspring, and it's just to try and make it so that you don't keep outcrossing with your relative, essentially. So I just pulled out the genes from our genomes from subs and cubes and just looked at what was going on. And in these pheromone receptor genes, we've got this perfect segregation of this particular gene and this particular gene. And the fact that there are two genes segregated meant that I was in over my head, because we've got more pheromone receptor genes than I expected, and they're segregating in the population. And this Saturday, I'll be talking to someone who knows more about Basidiomycota mating than me. At the very least, Psilocybe cubensis and Psilocybe subaruginosa are tetrapolar. So they have those four genders rather than two. So only 1 in 4 from a spore mass will cross with each other. And this is just the other locus, the Homeodomain locus. And this was the other surprise to me, was that it hadn't segregated in our population of 6. So this is still part of that same population. 208 is from the same population of subs that I'd collected. And really, that's a bit of a melon scratcher for me. So I'm looking forward, and hopefully I can give another update again later. I'd just like to thank everyone from the community who has helped me out, to these people, from the landowners themselves. Miss Bunyip, thanks for this specimen. This specimen itself is going to go into the herbarium. I think it's super cool. Even if you send me stuff that isn't a Psilocybe cubensis or a Psilocybe subaruginosa, if you send me the spore print, I'll definitely try and culture it. I probably won't culture Panaeolus cyanescens, because it just starts to get too much work, and the culture plates themselves are really expensive, and I get them for cheap because I'm in like a government building, but I just don't have the resources to keep it going because the project's not funded. Okay, yeah, these are the take-home messages. You can read them at your leisure, because I think I've gone on for too long already. Okay, and of course, it's all on the blog, but thanks, Emily. Bye for meow.
And thank you very much to Dr. Alastair Mctaggart, who presented there and has presented at a previous EGA webcast, which again you can find on the EGA YouTube channel. It's my pleasure now to introduce our guest this evening. Our guest is Dr. Jason Slott. Jason is an Associate Professor of Mycology and Evolutionary Genomics at the Ohio State University. Dr. Slott received a BA in Molecular Biology and Biochemistry and an MA in Science Education from Boston University. His doctoral dissertation at Clark University and his postdoctoral work at Vanderbilt University focused on the molecular evolution of ecologically relevant genes in fungi. As faculty of plant pathology at the Ohio State University in Columbus, Ohio, Dr. Slott uses comparative and functional genomics along with metabolomics to investigate the evolution and ecology of fungal chemistry in various fungal niches, including plant pathogens and symbionts, insect pathogens and decay fungi. Pioneering sequencing of psilocybin mushroom genomes in his lab recently led to the identification and evolutionary analysis of psilocybin gene clusters. Dr. Slott is a co-founder and scientific advisor for the Entheum Foundation, a non-profit for the science and conservation of entheogenic organisms. Welcome, Jason.
Thank you very much, Nick.
I believe you have a presentation for us this evening.
I do. So I guess I can just get started here. Let me pull up my presentation for you. And thanks for that great introduction. I am faculty at Ohio State University in the Department of Plant Pathology. And I'm going to pull up my slides here a minute. And so today I'm going to be talking to you about one project in our lab where we study the ecology and evolution of fungi. And this project is about what we can learn from the evolution of psychedelic organisms. In this case, we're going to be talking about psychedelic mushrooms. So in our lab, we study a lot about how fungi interact with other organisms. This panel here is showing you a number of different ways that fungi are interacting with other organisms. For instance, up here in the upper left, we have a plant pathogen, Cochleobolus, which is able to kill corn tissue to feed as a parasite. In the middle here, we have a mold, Aspergillus flavus, which is a major agent of corn and nut crop spoilage. Up here we have a lichen, which is a symbiosis between a fungus and an alga, and occupies very interesting environments, extreme environments, because of this symbiosis. And down here in the lower right, we have a mushroom that's actually a deadly mushroom. This is a deadly Gallerina. And then down here in the lower left, we have a representation of another type of fungus that interacts with human beings in a very interesting way, depicted by these figures, which appear to be somehow under the influence of magic mushrooms or psilocybes in the Mayan kingdoms of Mexico. Now, when I approach these systems here, I also think about the molecules that are involved. So that plant pathogen, Cochleobolus, produces this toxin, HC toxin, in order to be pathogenic to its host. And in here, this mold, Aspergillus flavus, produces a potent carcinogenic toxin, aflatoxin, which has to be monitored very carefully because of the destruction that it can cause livestock and the human food chain. Over here, we have a compound called usnic acid, which gives that light green color to this lichen here. And it's probably involved in protecting against too much feeding by reindeer, perhaps, or also protection from UV light. It's a bitter substance that blocks out UV light. And then down here, this is an alpha amatoxin. This is an extremely deadly molecule produced by a few species of mushrooms, and it's really a slow and painful death that it causes by basically preventing your body from reading its DNA with RNA polymerase. Really nasty chemical, which apparently has some sort of an ecological role to control the organisms that might feed on it. And then down here in the lower left here, we have psilocybin, which is the active constituent of a number of magic mushrooms. This is actually maybe more of the simplest of the molecules that are on the screen here right now. So I really look at these questions as, what are the molecules that are underlying the ecology of these organisms? And the case of magic mushrooms goes back thousands of years, we know, but it didn't really come to the attention of Western science until the late 1950s, when Gordon Wasson went down to Mexico and convinced Maria Sabina, a Mazatec shaman in Oaxaca, to allow him to partake in a magic mushroom ceremony. And his detailing of his experience in Life magazine in 1957 really led to the growth of the psychedelic movement in the United States and eventually in Europe. Gordon Wasson took those mushrooms out of their shamanic context, out of their mystical context, and brought them into the scientific context by sending some samples of Psilocybin mexicana to Albert Hofmann in Switzerland, who very quickly was able to isolate and identify the compound psilocybin as the active constituent of those magic mushrooms. And this is where somebody like me, had I been around 60 years ago and seen the publication of the psilocybin molecule, I probably would have been compelled to head down to Oaxaca and figure out, well, what's going on with these mushrooms and how did their psychedelic properties come to be, because I'm an evolutionary ecologist. Well, it wasn't me who went down 60 years ago. It was a lot of people exploring their minds, exploring their consciousness, the hippie generation, and also a Harvard psychologist like Timothy Leary went down to Mexico and also figured out ways that they could consume the magic mushrooms. And that really led to the expansion, the proliferation of psychedelics throughout Western society over the coming years. And it didn't necessarily have the happiest outcomes because a lot of the experiences that people were having were potentially very positive. They were giving people feelings of oneness, of desires for peace. And in the 60s and 70s, there was a lot of conflict going on in the globe, and the counterculture with the aid, I might say, of psychedelics really took to protesting the wars in Vietnam, the war in Vietnam and other places. And it really became a lot of conflict between the counterculture and Timothy Leary, who was leading a movement to expand your consciousness and throughout the country. And it really became an unsafe place for the military-industrial complex. And at the height of the protests of the war in Vietnam, 1971, Richard Nixon declared all non-alcohol, non-prescribed, non-tobacco drugs to be public enemy number one. And Timothy Leary was declared the most dangerous man in America. And this really set the stage for what we now know as the drug war. And it set the stage for how science of psychedelics proceeded over the next 40 years. So from the discovery of psilocybin in 1958, there was a growth of research, but then when the Controlled Substance Act was kicked in and psilocybin was scheduled as the most in the most dangerous category, Schedule 1, research really dropped to a trickle. And most of this research here was having to do with drug interdiction and testing, not necessarily for understanding the uses and biology of psilocybin itself. And then eventually, sometime in the early 2000s, we've entered what we call the Psychedelic Renaissance, where despite the regulatory status of psilocybin, a lot of scientists have gotten approval, and conducted new research to understand what it is that these substances are all about, and what can they do, and how do they work. And so we are now in what is called the Psychedelic Renaissance, And in this Renaissance, we're really looking at a lot of things about how the compounds in psychedelics are impacting the brain, both on a large scale and on the microneurological scale. And also, there's a lot of research that's been coming out over the past decade and a half of the applications and the potential therapeutic value from treatment for treatment-resistant depression to substance abuse problems. But at the same time, the basic understanding, the basic biology and ecology of these organisms has also lagged for the last 40 years, at a time when huge major advances have been happening in our ability to study living systems, from genomics to metabolomics and all these great new techniques that have come about, and we've really not had the chance to grow the field of psychedelic ecology, understanding of these organisms throughout those technical revolutions that have been going on. So I'm really interested in the ecology of these organisms and how they perform their ecological roles. And so psilocybin is produced by, in the big scheme, just a small number of mushrooms that are quite disjunct in their phylogenetic distribution. So we have a lot of lineages, or several lineages of unrelated fungi that produce psilocybin. And they seem to have, maybe some things in common in their ecology. So we have mushrooms like Psilocybe cyanescens, which seems to be really well adapted to mulching and landscaping in the urban environment. And we have mushrooms like Psilocybe cubensis, which is a dung decaying fungus, an herbivore dung decaying fungus. We have other dung decaying fungi like Paniolus species that fruit on hippopotamus and elephant dung. And there's a number of other wood decaying and even mycorrhizal Inocybe species. They have symbiotic associations with tree roots. So what is it about all these, this motley crew of various mushroom species with somewhat similar, but some also somewhat different ecological roles and evolutionary histories? What is it about them that makes them have psilocybin? So when we look at the phylogenetic distribution of the psilocybin compound, we've known this distribution for quite a few years now. This research had been done largely in the underground, but also in labs that could collect mushrooms and do basic chemical analysis over the past decades. And we found that psilocybin is produced by some very closely related species that are scattered across the phylogenetic tree of mushrooms, of agaricomycete fungi. For instance, a small number of species in the genus Gymnopolis, a larger number of species in the genus Psilocybe, Inocybe, Canocybe, it's actually Foliatina, and Paniolus. So what is it that causes them to all be so unrelated across this phylogenetic tree? Well, at this time, about 5 years ago, we couldn't answer that question because we didn't even know what the genes were that were responsible for making psilocybin, which is sort of the basics of understanding how a substance evolved, is understanding the genes that are involved in making it so that you can do evolutionary analyses on those genes. Well, we didn't know that. So our first step was to discover what those genes were that were involved. And we took a comparative genomics approach. We sequenced 3 magic mushroom genomes, Psilocybe cyanescens, Paniolus cyanescens, and Gymnopolis. This is now was changed to Gymnopolis dilipus. These 3 magic mushrooms were not very closely related. And then we chose for comparison mushrooms that were closely related to them that did not produce, or that at least we thought they did not produce, psilocybin. And we did a comparative analysis to screen for genes that were only in the psychoactive psilocybin producing fungi and not in their close relatives. And then we looked for gene functions, and we looked for what we call gene clusters, and I'll show you this in a minute. And it turns out that there was one cluster of genes that distinguished psilocybin producing fungi from psilocybin not producing fungi. And so you can see that represented here. We have a couple of mushroom species that are related to Psilocybe cyanescens. This is Gallerina Marginata. And when you line up their genomes, this is a section of their genome, this chromosome here. There's genes in a particular order. And in the psychoactive Psilocybe cyanescens, you can see that in this position here, where it lines up with Gallerina Marginata, there is this cluster of genes. And it turns out that this cluster of genes is responsible for the production of psilocybin. This was the functions of all these genes were determined in parallel in Dirk Hofmeister's lab in Jena, Germany. And basically it comes down to 4 enzyme encoding genes here, and there's a transporter gene involved as well, which the function we don't know yet. So getting back to our question, why do so few of these fungi produce psilocybin, and why are they so recently distributed across the phylogenetic tree, and why are they so dispersed across the phylogenetic tree? And it turns out in our work and in a follow-up study by Awan et al., it turns out that there were multiple horizontal gene transfer events where that entire cluster was copied from either Gymnopolis species or Psilocybe species. It was copied into another lineage altogether and then proceeded to diversify amongst the species in those alternate lineages. At least 3 horizontal gene transfers have been identified. And then there's also the suggestion from the Awan et al. Paper that there may have been an independent evolution of completely different genes forming a completely different cluster in the mycorrhizal lineage Inocybe. And when we take a closer look, if we zoom in to the phylogenetic trees, the evolutionary history of some of these genes, in this case, we're looking at the tryptophan decarboxylase gene, side E. Acid tryptophan. When we look at the evolutionary history of that gene, we see that the horizontal gene transfer that happened from the lineage Psilocybe to Panaeolus really seems to be associated with fungi that are breaking down dung. So this kind of gives us the clue that the dung ecosystem provides the selective pressure to acquire and maintain this particular function to produce psilocybin. It also turns out that some of the genes that are in this cluster have an enriched distribution amongst dung fungi to begin with. So something about the dung niche seems to be potentially important for the production of psilocybin. But what could that be? So the dung niche is kind of novel. We haven't had mammalian herbivores on Earth forever. We've had, since the KT extinction boundary 65 million years ago, when mammals sort of hit the stage and started diversifying into a number of niches, there was also a decline of forests and an expansion of grasslands over the ensuing tens of millions of years. And there's really the major grassland expansions happened in the last 20 million years through the Miocene period. And at the same time that those ecological changes were happening, fungi were adapting to the novel environment. So Psilocybe can be inferred to have had an ancestral state of being a wood decay fungus, probably small bits of wood and litter debris, also maybe fallen logs, that sort of environment. But as Psilocybe diversified in this environment that was gaining more grasslands and more open bare soil, Psilocybe adapted to these niches, and some fungi became disturbed soil growers. Some of them grew on those grasses. And then eventually you see arising a lineage that decays dung, because you've got these giant herbivores, mammalian herbivores, that evolved eating large amounts of grass and producing large amounts of herbivore dung on the grasslands. So this isn't a unique story. This also happened in other lineages like Panaeolus, that had a sort of a wood and litter decay ancestry, and then eventually diversified to adapt to the expanding grassland niches, and eventually to herbivore dung as a source of food. So what is it about that dung niche that selects for psilocybin? We're still scratching our heads about this question. And we thought maybe if we can identify the origin of the psilocybin gene cluster, we might get some more clues about what the psilocybin is doing in the environment, doing ecologically for the fungus. So this is another phylogeny of that Cy-D gene, the tryptophan decarboxylase. And you can see these are the gene clusters we've been talking about in the agaricales and these mushroom lineages. You can see there's a couple of horizontal gene transfer events that we were just talking about, one to Panaeolus, one to Foliatina. And then this set here is the only part of the tree that we were able to identify gene clusters that make the production of psilocybin possible. And the closest relative of those genes was found in this lineage here, this surprising result in this crust-forming fungus lineage, which contains Fibula rhizoctonia. Now, this fungus here in this tree is actually a carrot pathogen, but what got us really excited and interested is that it's closely related to this fungus here. Now, you might not see a fungus here. This is Fibula rhizoctonia that makes what they call termite balls. So these little spheres here have the same diameter and the same outer texture as the eggs of termites, of Reticulotermes termites. And they somehow manage to convince the termites to take care of them in piles of their own eggs. And it's called the cuckoo fungus for that reason. Cuckoo is a bird that lays its eggs in other birds' nests, and the host parents raise the cuckoo for the cuckoo parents. So in this case, this fungal sclerotium here, this resting structure, is cared for and eventually germinates in the retired brooding chambers of these termites and carries on. So we thought, hey, maybe something to do with psilocybin. Maybe the psilocybin molecule could be involved in manipulating these termites to take care of these as their own eggs. So we sequenced the genome of the termite ball fungus here, and then it turns out that they only had 4 out of the 5 genes themselves, whereas the carrot pathogen had at least 1 copy and sometimes dozens of copies of the psilocybin genes. The only other species that we found that had all the genes required to make psilocybin, but the termite balls fungus, the cuckoo fungus, was missing that final step, the psilocybin synthase gene. And we also followed this up with metabolomics, and we determined that the cuckoo fungus does not make psilocybin. So we kind of ended up at a dead end here. But we still think that this lineage, this Athelia lineage, could be the origin of psilocybin. But it's also possible that this lineage got the genes for psilocybin synthesis and then eventually started using them for something else. So we really haven't nailed down the origin of psilocybin, which would be really nice, because if we can find the origin, we might be able to find the center of diversity for this type of biochemistry, which might give rise to finding novel analogs of psilocybin, novel neuroactive compounds potentially. So currently, this is our model for the evolution of hallucinogenic mushrooms. In the bigger picture, this is a bigger scope of Earth history. So hundreds of millions of years ago, we had wood came to dominate the landscape. And wood is a real problem for other organisms on Earth. It's not a great food source. It's impregnated with lignin. It's a lot of cellulose impregnated with lignin, which is hard to chew through. It's hard to digest enzymatically. And really, a couple 100 million years ago, I believe is the date now, the only things that could degrade any appreciable amount of wood were wood decay mushrooms and the agaricomycetes. So they really had a monopoly on decaying wood for millions or tens or hundreds of millions of years until about maybe 100 million years ago, a lineage of cockroaches evolved that we now call termites. These wood-eating insects come onto the stage, and they are competitors for decaying wood. They'll be interacting with wood decay fungi, and this really would set the stage for competitive biochemical interactions between mushrooms and insects. So we think that maybe those early interactions were the impetus for the evolution of psilocybin, the origin of psilocybin in wood decay fungi as possibility. We can trace psilocybin back to maybe between 25 and 70 million years ago. And then we had this KT extinction event 65 million years ago, which wiped out, a large portion of prior species on Earth. And then you had the emergence of mammals. And within 5 million years or so, those mammals that were initially very tiny had grown to dominate the landscape as these large megamammals, largely herbivorous megamammals, and leaving lots of large herbivorous megamammal dung on the environment. It opens up the niche for wood decay fungi to transition into dung decay fungi. So now we have psilocybin producing mushrooms in the dung decay niche, which sets the stage for dispersal amongst different mushrooms in the dung decay niche. So again, we come back to what is it about the dung? Well, we suggested that it was really the other organisms that are living in dung and maybe also in the really rotten wood and wood chip type environment that have direct access to mycelium of fungi. Fungal tissue is really a good nutrition source for invertebrates like nematodes and insects, arthropods. There's a lot of fungus-eating invertebrates out there. And so fungi like the dung decay fungi are competing with a bunch of these organisms, from fly larvae to dung beetles to termites, and they could be really under a lot of pressure to delay the feeding rates of these organisms. And perhaps the activity of psilocybin on 5-HT2 receptors, which are conserved throughout the animal kingdom to some extent. Insects have the same receptor for psilocybin, at least the same family of receptors as humans do. Perhaps it could be deterring the feeding, reducing the feeding on fungal tissue to produce psilocybin. Well, then in 2018, we got this other clue that psilocybin could be targeting insects. When Cassin's lab at Western Virginia University identified psilocybin in the plugs, the fungal plugs on these infected cicadas, these Mesospora infected cicadas. And if you're not familiar with this story, when these periodical cicadas emerge after 18 years under the ground, as they're emerging, they can be infected by a pathogen called Mesospora. And it very quickly, in a very short period of time after the emergence, it colonizes and replaces the abdomen of the cicada with this reproductive structure here, this sporangium. And you can see there's like a dust here. The cicada actually stays alive throughout this entire process, and it flies around and it dusts these spores. And not only does the cicada fly around, but it's hyperactive and it's hypersexual. So the fungus is altering the behavior and the sexual spread of the disease somehow. And the idea that there's psilocybin potentially involved is very interesting. This is a very difficult system to study. It hasn't been proven that psilocybin is responsible for this behavior, but it's certainly an interesting hypothesis. It's not the only hypothesis for the ecology of psilocybin. There's another hypothesis that relates to the bluing reaction. So a lot of magic mushrooms, a lot of psilocybin-producing mushrooms stain blue when they're injured or when they're bit by another organism. And that blue stain, Dirk Hofmeister's lab recently showed, was the result of polymerization of psilocin. And when psilocin polymerizes, it reflects light differently and produces this blue color. But that those polymers are thought to perhaps mimic the tannins that plants produce as antioxidants. And so there's an alternative hypothesis that psilocybin is not necessarily even serving a neuroactive role in its ecology, but maybe just a disruption of feeding in general. However, there's a lot of diversity in the effects of mushrooms that make psilocybin. This is a woodlover species, Psilocybe cyanescens, I believe. And the woodlovers seem to have a little bit more effect than the psychedelic trip or the reduction of feeding by something that consumes it. They also have been reported to cause a temporary paralysis known as woodlover's paralysis, which could suggest that there's some diversity in the chemistry, the neuroactive chemistry of psilocybin mushrooms beyond what we already know about. And this is one of the places that Entheome, which is a nonprofit organization organizing the Entheogen Genome Project, where they come in. And this is a project that is seeking out as our first mission to basically generate reference genomes for all the magic mushrooms. We're targeting 140 psilocybe, 20 gymnopolis, 20 paniolus, and 4 Canosphere foliatinus species. This is a crowd-funded open data initiative to really just flesh out the genetics and the biodiversity that is out there of magic mushrooms and other entheogenic species eventually, for the purposes that we're all interested in. We're interested in preserving the species, the genetic resources, and hopefully the environments that are the home to these organisms that set up their ecology in the first place. So the Entheogen Genome Project has started to generate some genomes, and in the earliest results, we're actually starting to see some important clues. The first clue that I'm going to show you actually has to do with this gene here, Psy-H. This is a hydroxylase of the 4 position on tryptamine, which is actually a really somewhat rare biochemical reaction to be found in nature. There's another fungus, ergot, produces a 4 substituted tryptamine backbone and leads to the production of ergotamines, including lysergic acid, the precursor of LSD. As we are sequencing these genomes, we're pulling out the gene clusters and the homologous genes of psilocybin biosynthesis pathways. And what we're finding is that there's quite a bit of diversity in the gene clusters. And particularly what we're noticing is multiple copies of the Psy-H gene. And those multiple copies have different structures as well, indicating that they could be taking on new functions or somewhat related functions. Now, what those could be, we don't know. These are initial clues. Perhaps they could be producing analogs and parallel pathways to the production of psilocybin. They could be doing something completely different. But the fact that they're usually still residing in these gene clusters and they have evidence of their structure having changed makes us think that perhaps there is maybe some analogs produced by some of these alternate gene clusters, psilocybin analogs. And this is a phylogeny of some of these Psy-H genes here. And yeah, you can just see where they appear and have generated these long branches, which suggests that they've gone off on a new evolutionary trajectory. So to wrap up, these are the questions that we're currently working on, which I've sort of introduced you to. When and where did psilocybin originate? That might give us a little bit more clues as to what psilocybin is doing in the environment. And then, of course, as we sequence these genes and these genomes, we can start to identify maybe new undiscovered analogs of psilocybin that could have similar functions or even novel functions. Psilocybin has been shown to have not just psychoactive properties, but also neuroplasticity properties and perhaps even other therapeutic uses. So discovering analogs does have inherent value for people. And then finally, what's maybe closer to my main interest is what can these genomes tell us about the evolution of psilocybe, ecology, as it goes through time and adapts to these various niches? How can we understand the genetics behind fungal adaptation, fungal ecology, and evolution? So I will end there with just thanking all the people that have been involved in this research that I've been working with and associated with, and some of the services that have also played an important role. And I'll open it up to questions if you have them.
And thank you very much, Jason, for that presentation. We do have some questions, and thank you to everyone who has dropped a question into the chat so far, and I'll get you to drop out of screen share, Jason. If you do have a question, do drop it into the comments section, and we will get to it as soon as possible. But to start us off, how common is HGT between fungi species coming from Darklight? Thank you, Darklight.
That's a great question. From my perspective, it's fairly common. But I think in terms of evolutionary time, which is in millions of years, tens, hundreds of millions of years, and so if you were to compare it to horizontal gene transfer in bacteria, it's fairly rare. It's at least rare that it sticks, that we identify it. In any given lineage, you might find any given fungus, you might find that 5% of the genes of a mold, for instance, have been recently horizontally transferred into that fungus. Whereas if you look at a bacterium that you isolate from a hospital, that number may be 50, 60, 70% recently horizontally transferred genes. And that horizontal transfer in bacteria happens on a daily basis. So I would say that it's an important part of the ecology, but it's not happening constantly every day.
Darklight is also wondering if they can have a copy of the PowerPoint presentation. I suspect that's something that we can handle afterwards, and again, make sure that you are signed up for the EGA updates. But I suspect it's probably yes.
Yes, I can do that.
Our next question comes from Emma. Is there HGT in conserved regions?
So I'm going to interpret that question as, are there specific parts of the genome that are more likely to have HGT? And I would say that the answer is yes-ish. There are certain regions of the genome that evolve more quickly. We think of these as the regions adjacent to the ends of the chromosomes, the subtelomeric regions. They seem to accumulate a lot of changes, including horizontal transfer events, because they have the activity of a lot of transposable elements which rearrange the genomes. And maybe the more stable parts of the genome are in the center of the chromosomes, as opposed to the ends of the chromosomes, which evolve much more rapidly.
This is a question from myself, actually. As we were going along and you read about I think it was the 50 to 75 million years ago when the insects came into the evolutionary process, you did mention later on that you hypothesized that there isn't a neurochemical role of psilocybin in the interaction with the insects, but I wasn't sure if that was at that case of 50 to 75 million years ago. So was there or is there any understanding of there being a neurochemical role that influenced the evolutionary path at that stage?
We have no direct evidence of a neurochemical role of psilocybin on insects. We have circumstantial evidence. We know that other 5-HT2A agonists can impact the feeding rate of fly larvae, for instance. And we know that there are specific ways that can happen by down-regulating neurons in the esophagus of insects. But the actual research on psilocybin in insects itself is nearly non-existent. There's older research that has given psilocybin to a number of insects, but it was really just monitoring behavior and seeing that, yes, in fact, it can alter the behavior of spiders, for instance.
At the start of your presentation, you noted the Psychedelic Renaissance, which is just going on 15 years old now. What are some of the pros and cons of especially in the past 5 years, the psychedelic industry boom on your research?
Well, people have certainly taken more of an interest in really only the past couple of years. I think the majority of the attention has come from the clinical side for a lot of this. I mean, it's for obvious reasons, people are interested in psilocybin for its potential use, benefit, possibilities for human beings. And my research is a step away from that. And while our research can potentially be of use to identifying novel psychedelic compounds, that's not what has been at the forefront of people's mind until much more recently. But as the industry is growing, people are looking for new niches and new ideas for businesses, and it's definitely created more of a role, more of an interest in what the mycologists can contribute to the field.
Our next question comes from Kane, who I noticed is also part of the Entheome Foundation. You can find the link for that in the scrolly box below, entheome.org, I think it was. Would it be possible to set up an experiment to try to observe HGT in action? And how would we go about tracking it, and how stable the transfer was?
Yeah, I mean, it's certainly possible. These experiments are routinely done. We use horizontal gene transfer potential mechanisms when we're doing any kind of genetic engineering or genetic modification. If you want to do transgenics, we're using evolved mechanisms that we've turned into techniques to transfer transfer genes. And we basically just force the process of evolution to happen in a test tube, right? But what's happening naturally is a different question, right? If something horizontally transferred 25 million years ago between two mushrooms, can we really recreate that exact environment and to know exactly why it naturally happened? Or are we just forcing something that can potentially happen in laboratory? So we have clues of the natural mechanisms, but we can't necessarily easily observe those natural mechanisms in action. But we can certainly force it to happen by putting two fungi together, giving them some stress. One of the fungi will pick up DNA at some point, and if that DNA has some sort of means of providing a selection, then we can select for the fungus that acquired that gene, and there we've demonstrated horizontal transfer. But is it real? Is it a demonstration of a natural process? We won't know.
This one from Osip Off Photo, and if you do have a question, do drop it into the comments section. Does psilocybin affect dung producers as well? I think you sort of answered that before, but is it different for the dung producers and the coprophilic mushrooms?
So affect dung producers as in the herbivores, I'm taking that to mean. I think the psychedelic mushrooms are really not doing their thing in the animal. There's a chance that the mushrooms come up out of the dung and a cow will come by and eat the mushrooms, but it's not as likely that it's going to, because animals have an instinct to avoid their own feces for disease prevention, evolutionary reasons. So chances are, in my opinion, they're not having any effect on the herbivores themselves and the animals that produce the dung, because the spores really don't germinate and get going until the dung has already left the animal's body.
I don't know if there would be an answer to this question, but is it just pure coincidence that that molecule psilocybin happens to be active in human species and I suppose other species with similar neurochemistry? Is that a pure sort of natural coincidence, or is there another explanation for why that happened?
I mean, I would be open to multiple hypotheses here. I think initially, if you look at the time frame over which psilocybin probably evolved, I think initially it was probably driven by invertebrates, which have a large biomass, which are large pressure on fungi directly. But that's not to say that there weren't mammals involved at some point in selection for psilocybin. There are, in these grasslands, there's also voles and mice that would feed on mushrooms, and they could pose a strong selective pressure on the reproduction of dung fungi. So it's possible that while it was probably initially driven by invertebrates, that mammals, probably not people, which only evolved extremely recently, but mammals themselves may have been a secondary target for psilocybin.
And further to that question, this one from Melissa in the comments section, asking about the stoned ape hypothesis from the Mckenna brothers. She says she's heard of it but not done much reading on it, and is curious to know your thoughts on this, and whether or not there's any evidence for it.
It's a great question, and I think it's opened up a lot of evolutionary thinking in the psychedelic community, and I'm grateful for the stoned ape hypothesis for that. The stoned ape hypothesis has been criticized for the evolutionary mechanisms that were suggested that are not necessarily supported by evolutionary studies. So I think I mean, a lot of pieces of the hypothesis could be well argued, but that magic mushrooms, really drove the evolution of humans. I think you need maybe a different model, and I think that there's a recent paper by Rodriguez Arce and Winkleman that talks about psychedelics in the sociocognitive niche, which I think is really the right area to be looking at this question. It's so where Mckenna was talking about the immediate visual acuity that may have given hunters an advantage that may have selected for the people who were consuming magic mushrooms, that really would lead to maybe more of a dependence on magic mushrooms amongst people rather than an actual physiological change in brain anatomy or visual anatomy or something like that. Whereas yes, that's it, the psychedelic sociality and human evolution. What this is positing is that psychedelics could have been involved in human evolution by playing a role in the sociocognitive niche, which is what humans have in particular. We're sort of freed from most of our ecological constraints to our reproduction and food gathering, and we're really driven by the niche that we build for ourselves, our civilization, our technology, our extrasomatic knowledge. All these things are really our current niche, and psychedelics feed into that by changing our ideas and helping us visualize new technologies. And it's these technologies and art and cultural changes, these are the things that really have been driving our evolution for hundreds of thousands or even millions of years. So I favor looking in that direction, inspired by the Stoned Ape hypothesis, but thinking in a more sophisticated fashion going forward about the sociocognitive niche.
I just posted a link to that article into the comments section as well if you want to have a read of that one. We have a few questions left. This one from Darklight. Will Entheome extend its focus beyond the fungal into plants as well?
The plants are certainly on our radar. We really want to do a good job on fungi first, and there will be other people interested in sequencing entheogenic plants. There are a couple of entheogenic plant genomes out there now, and so I think we'll be surveying what that landscape looks like in the coming year or two, and we'll see how we get into that world, plants and animals. I don't think there's any reason to leave out the conservation directed research of entheogenic animals as well.
Well, thank you, Jason. Unless anybody has any final questions, then we really appreciate having you here. I know that you've called in from the UK, and it's very early in the morning there, so we appreciate you getting up so early to present this fantastic piece of information and the research to us today. And I'll just have a look quickly. Looks like there's no other questions. If you have any final comments, maybe anything on your future radar in the next 6 to 12 months that you might like to share, I would love to hear from it before we finish up.
Great, and thank you very much to Entheogenesis and you, Nick and Kane, for having me. Yeah, I would just hope everybody looks into the Entheome Foundation. I'm also at The Ohio State University in Ohio, and we're holding the second Psychedemia conference. The last Psychedemia happened 10 years ago at Penn State, and now it's happening the end of this summer at Ohio State. So take a look at that, and maybe you'll want to attend there. And other than that, thank you.
Thank you very much. And I think I just found the Psychedemia website. This looks like the one. So I will drop that into the chat as well. Really appreciate. And you're in the US, not the UK.
That's correct.
Yes, sorry. In the US, it's still early in the morning, so really appreciate that, Jason, and thank you very much for being with us today.
Great, thank you.
And that was Dr. Jason Slott, Associate Professor of Mycology and Evolutionary Genomics at the Ohio State. I keep going to say Ohio State University. Dr. Slott had his dissertation at Clark University, postdoctoral work at Vanderbilt University focused on molecular evolution of ecologically relevant genes in fungi, and we thank him very much. Thank you for all of your comments and the lively chat as well in our comments section today. If you haven't already, make sure to subscribe to our YouTube channel and make sure to share it as well with anybody who might be interested in EGA and the work that we do here. Again, I'll just quickly mention Garden States, of course, coming up at the end of the year. Make sure that you sign up or register your interest at the EGA website. And I'll just head across to the EGA website. Here we go. Again, on the front page there, click here to register, take you to the registration form where you can register your interest. Not selling tickets as of yet. If you are a current ticket holder, of course that will be valid for the end of year conference, but we are keeping a close eye on the COVID situation just in case, because it's thrown us a lot of curveballs over the many months, years now of this situation. And of course, next month as well, let's go back, we will be hearing from the team from Mardi Gras, who will be talking about the Mardi Gras Protestival, which is now happening in September. That has been postponed due to the flooding that's going on right now in the northern rivers, and it's affected a lot of people. So again, our love, compassion, our thoughts out to everybody there who might be tuned into it, into this right now or at a later date. We hope that things are rebuilding quickly, and we hope that incidents like this aren't irregular, but sadly it is looking like that will be the case. Again, that's the chart, the comparisons chart, which will be the ethnobotanical combination chart from EGA, and there will be an NFT available for that once it's released as well, and that will help to raise funds for EGA for the fantastic work that it does. The resources at EGA, entheogenesis.org, a growing list of resources there, and also on our YouTube channel where a video was just put up today of Kane talking about the psilocybe species, and you can find that by heading to the videos there. And there's Kane, an introduction to cubensis, and you can find that on our YouTube channel, and there will be more videos added over the coming weeks, so make sure to keep an eye out. I'm actually not too familiar with this, but I believe there's some kind of bell. Oh yes, there you go, up in the top corner there. You click that bell and you'll get notifications on your YouTube, as long as you're signed into an account, when new videos are released, when live videos are available, when there's action from the EGA YouTube account. So make sure to do that. And stay connected with us, entheogenesis.org/connect. Thanks everybody for tuning in today. My name is Nick, and we will be back in April. I hope you enjoy your autumn. It's certainly getting cooler. The leaves are changing colour, and soon enough, I suspect, at least around Victoria, we'll be seeing a lot more fungal growth as well. Thank you very much, everybody. Enjoy your evening. Bye-bye.